The difference in their size is due to the asymmetry of the deoxyribose rings and the structural distinct nature of the upper surface of the base pair relative to the bottom surface. Thermal properties of DNA. If a solution of DNA is subjected to high temperature, the H bonds between bases become stable, unstable, and the strands of the helix separate in a process of thermal denaturation. Regions of the duplex that are predominantly AT base pairs will be less thermally stable than those rich in GC base pairs. Right. DNA molecule exists as single strands. This point is the melting point, melting temperature Tm, and is characteristic of the base composition of the DNA molecule. The Tm depends several factors in addition to the base composition. These include the chemical nature of the solvent and identifies the concentration of ions in DNA molecule. The Tm depends on several factors in addition to the base composition. These include the chemical nature of the solvent and identifies the concentration of ions in the solution. DNA, the genetic material, the physical carrier of inheritance. Frederick Mason in 1869 isolated DNA from fish sperm and the pus of open wounds. Since it came from nuclei, Mason named this chemical nuclein. Subsequently, the name was changed to nucleic acid and lastly to deoxyribonucleic acid. Robert Fulton in 1914 discovered that function-distained DNA was then found in the nucleus of all eukaryotic cells. During the 1920s, biochemist Pierre Levine analyzed the components of the DNA molecule. He found it contained 4 nitrogenous bases: cytosine, thymine, adenine, and guanine, deoxyribose sugar, and a phosphate group. He concluded that the basic unit nucleotide was composed of a base attached to a sugar and that the phosphate also attached to the sugar. He unfortunately also erroneously concluded that the proportions of bases were equal and that there was a tetranucleotide that was repeating structure of the molecule. The nucleotide, however, remains as the fundamental unit monomer of the nucleic acid polymer. There are 4 nucleotides: those with cytosine, those with guanine, those with adenine, and those with thymine. During the early 1900s, the study of genetics began in earnest. The link between Mendel's work and that of cell biologists resulted in the chromosomal theory of inheritance. Garrett proposed the link between genes and inborn errors in metabolism, and the question was formed, what is a gene? The answer came from the study of deadly infectious disease pneumonia. And during the 1920s, Frederick Griffith studied the difference between a disease-causing strain of the pneumonia-causing bacteria Streptococcus pneumoniae and a strain that did not cause pneumonia. The pneumonia-causing strain, the S strain, was surrounded by a capsule. The other strain, the R strain, did not have a capsule and also did not cause pneumonia. Frederick Griffith, 1928, was able to induce a non-pathogenic strain Frederick Griffith was able to induce a non-pathogenic strain of the bacterium Streptococcus pneumoniae and to become pathogenic. Griffith referred to the a transforming factor that caused the pathogenic. Griffith injected the different strains of bacteria into mice. The strain killed the mice, the R strain did not. He further noted that if heat killed S strain was injected into a mouse, it did not cause pneumonia. When he combined heat killed S with live R and injected the mixture into a mouse, remember, neither alone will kill the mouse. That the mouse developed pneumonia and died. Bacteria recovered from the mouse, had a capsule, and killed the other mice when injected into them. The hypothesis: the dead S strain had been reanimated, resurrected. The live R had been transformed to live S by some transforming factor. Further experiments led Griffith to conclude that number 2 was correct. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarthy revisited Griffith's experiment and concluded the transforming factor was DNA. The evidence was strong but not totally conclusive. The then current favorite for the hereditary material was protein DNA and was not considered by many scientists to be a strong candidate. The breakthrough in the quest to determine the hereditary material came from the work of Max Delbruck and Salvador Luria in the 1940s. And bacteriophage are a type of virus that attacks bacteria. The virus that Delbruck and Luria worked with were those attacking Escherichia coli. A bacterium found in human intestines. Bacteriophages consist of proteins and a coach covering DNA. Bacteriophages infect a cell by injecting DNA into the host cell. The viral DNA then disappears while taking over the bacterial machinery and beginning to make new virus instead of new bacteria. After 25 minutes, the host cell bursts, releasing hundreds of new bacteriophages, having DNA and protein, making them ideal to resolve the nature of the hereditary material. DNA. In 1952, Alfred D. Hershey and Martha Chase conducted a series of experiments to determine whether protein or DNA was a hereditary material. By labeling the DNA and protein with different and mutually exclusive radioisotopes, they would be able to determine which chemical DNA or protein was getting into the bacteria. Such material must be the hereditary material, Griffith's transforming agent. Since DNA contains phosphorus, P, but no sulfur, as they tag the DNA with radioactive phosphorus 32. Conversely, protein lacks P but does not have S, thus it could be tagged with radioactive sulfur 35. Hershey and Chase found that the radioactive S remained outside the cell, but the radioactive P was found inside the cell, indicating that DNA was physical carrier of heredity. And polymorphism in DNA. A form having 11 base pairs instead of 10 base pairs per turn. The base pairs are not perpendicular to the axis but are tilted. C form like B form but having 9 base pairs. Per turn. D form like B form but have 8 base pairs per turn. Z-DNA, DNA with left-handed coiling is called Z-DNA. DNA supercoiling. A supercoil is when the double helix twists around itself, right? Supercoils can be positive or negative, but DNA exists in the negative supercoil form. DNA can be supercoiled if it is circular or if it is linear. And as fixed ends, supercoiled DNA is more compact than relaxed DNA. Negative supercoiled DNA molecules are easier to unwind than relaxed molecules. DNA unwinding is required for replication and transcription. Excuse me. Topoisomerases are enzymes that catalyze changes in DNA supercoiling. Right, and type 1 topoisomerases function by breaking a phosphodiester bond of one strand, passing the other strand through the break and resealing the break. They can only remove supercoil and increase linking number by 1. B. Type 3 topoisomerases function by breaking both strands and passing a double strand region through the break before resealing the break. Before this process, they require ATP. They decrease the linking number by 2. Topoisomerases are targets of numerous chemotherapeutic drugs: Erythromycin, VP16, tenopside, VM26, etoposide, and camptothecin. Hyperchromic effect. On denaturation of dsNA into S's, DNA UV absorption at 260 nanometer increases. 18. Packaging of DNA in eukaryotes. DNA is packaged in the nucleus of a nucleoprotein called chromatin. Levels of chromatin packaging for nucleosomes. Nucleosomes coparticles consist of 140 BP of DNA wrapped around a protein octamer. It's called a squabble 140 BP, consisting of just 2 subunits of histones, H2A, H2B, H3, and H4 linker DNA. Squabble 60 BP is the DNA between 2 core particles. H1 binds to the linker DNA and the core particle. B 30 nanometer fiber solenoid. Nucleosomes are wound into a solenoid-like structure. It requires histone H1 binding to every linker DNA. Chromatin C. Chromatin is a template for replication and transcription and substrate for DNA repair and recombination. So that brings us to the end of this lecture. I'll be continuing with nutrition and vitamins in the next lecture. Make notes, take your screenshots, And if you have any queries, come back to me on the comment section or come back to me on the WhatsApp section. I'll be more than happy to answer your queries. Until then, best wishes. Keep a schedule of learning, keep a timetable, keep to it, work, play, do everything together, but don't miss up and mess up. You should be okay. You should be fine.